Hello. I can't hear you. No.
Bengi challenges kibao. Still can't hear me? What is it? Hebu ita Audrey aniangalilie hii ya mute yenye naona. Audrey, we can hear you. We can now. Very good. Very good. We can now hear you. Very good. Sorry for the disturbances and yeah. You know, I'm not Gen Z, so I'm still experiencing all these challenges. So I tried the YouTube. I couldn't upload file. Even now, I still don't see the icon for uploading file, but one step at a time. But I'm happy that you can hear me now. Please, let's log in. Thank you so much. Let's log in. So I'm looking for a place to click so that I can upload the file for the questions. But I can't see it. But we are still uploading. Please upload. Come. Welcome. Because I have another option. I'm going to read the questions, each one of them, carbohydrates, proteins, lipids. So we need to have a pen and a paper. If the upload side, I don't see it. I'm going to read each and the questions. You need to have a paper and a pen and tell you how we need to answer such questions whenever you see them. Because the challenge which I had was, I don't see the icon for uploading the file. I'm still new in YouTube and new in Google Meet. I'm always used to Microsoft Teams, but I'm happy that you can hear me. Please, let's log in. The people who are on the other side, let them come this side. I'm sorry for the inconvenience caused. We are still at 40. We are waiting for more so that we can start. So if somebody has a clue how I can upload the file, you can tell me. You know, we have people who are gurus. They can tell me. Get my laptop. I'm going to read the questions now. Do somebody have a clue why I can upload a file? Because I need to upload the file. It's not like a file, but it's slides which shows different questions. And I'm going to send it as well to the channel after this. But what I want each and the students to have is you have a paper and a pen. We are going to discuss each of the questions just as they appear on the PowerPoint. If I miss to get a place for uploading, so somebody can tell me, because I don't see where to upload a file. I need to upload a file. Oh, Patrick, you are saying I can't upload any file for YouTube. I didn't know. So that is good. Everyone, you need to have a paper and a pen. I'm going to read the possible questions and I'm going to explain how we need to answer them. Thank you for that, Patrick. We are going to take our time. Any of those questions, whenever you see them, what you need to answer, very important, very important. Let us keep on logging in. Let us keep on logging in. And then you'll tell me maybe by 9:20 we shall start. Let people come, keep on logging in, because we can't wait forever. So we'll start as they try to find us as we proceed, because we need to discuss the carbohydrates, the possible questions, the lipids, the possible questions, the proteins, the possible questions, and a bit of buffers and water. So I thank you all. Keep on logging in. I'm seeing we are at 55. And this one, people can automatically come in, I think. After sharing the link, Asfa and Jennifer, kindly share. Good. 15 people have already come in. We are at 65, a very good number. At 100, I think by 9:20, we'll be at 100. Please have your paper and a pen. I'm going to read the questions. Carbohydrate. I read one, I explain what you need to answer, how you need to answer it. The second one, that way, until we finish all the questions. Patrick has helped me with an idea. He has said, oh, Peter, good, you are present there. Hello. Let us keep the spirit. Oh no, Akai need somebody needs the link. My weekend students, BMLS, we are all combined here. My nursing students, they are here. Are they here? I did share with them. So as we log in, let me try to share with them. Let me try to share. If they'll find, then they'll be able to log in. So let me see. Thank you so much. Thank you so much. Let me see how I can share with my students. And somebody forward it. Some of my students are not there. Good. Nakaya, very good. Kindly share with the other ones. I'm sorry for not sending it to you. I had a lot of challenges. So I already thought I sent only the MBCHB. Good. Sheila, very good. You are there. Please log in. I need to send to my BMLS students. Let us keep on logging in. At 100, we shall start. Please, let us have a paper and a pen. Patrick has just told me, Madam, you can't upload anything. Good evening, Madam. Kimathi Morin. Yes, hi. Oh, somebody can't hear. Simon, Wiek, Kwok, can you check your settings? Can you check your settings, please? Wiek, Kwok. You can log out and log in again. Oh, thank you. Thank you, everyone. We are here. Together, we shall achieve it. We shall pass. All of us shall pass. Let me try to share with the weekend group, BMLS. As we log in, we are at 75. Abdul Latif, Mohammed, good. We are here, madam. Is that the BMLS? Abdul Latif, is it BMLS? Please just tell me BMLS or BSCN or MBCHB, then I can tell you to share with Wakina Stella. Good, good. Abdul Latif, kindly share with Wakina Stella. Not in a bad way. I had a lot of challenges. Thank you so much. Thank you so much, BMLS. Thank you so much, BSN. Thank you so much, MBCHB. We are almost starting. Kindly have a paper and a pen. I was having challenges. I had challenges on uploading. Somebody has just chanwaring me, telling me, Madam, you can't upload anything on YouTube. But I don't know. But at least a step. Hata mtoto anaenda pole. We shall achieve. Good. Keep Benson, BMLS, you are there. Thank you so much. Let us keep on logging in. We need to start by 9:20. We were giving them like 4 minutes because I started with Google Meet. So it will be unfair if I start before many people log in YouTube. Thank you so much. I can see, Kaume is there. Good. Please just say BSN, BMLS. Madam is saying hi. We are almost starting. So have a pen and a paper or a book because I'm going to read each and every sections as I had them on my slides. I separated the carbohydrates. I separated a bit of water and buffers. And for the nursing and MBCHB, I've just uploaded how to derive the pH of water as 7, just some minutes back. I'm going to upload nucleic acids for the BMLS, for the BSEN, for the MBCHB. It doesn't mean that a question can't come there. A question can come anywhere, whatever we've been taught in class. So I've done the buffers, a bit of introduction on buffers for the students who have seen the channel. That is for the BSN and the MBCHB. Not for the BMLS. Please don't crack your head for that. I didn't teach it, so it is not going to be there in any of the exam. But even for some questions like enzymes, we are going to revise generally. The MBCHB do not do enzymes, but BMLS and BSN do enzymes. So we are just going to tackle how we can be able to answer a question when you see it, so that you can scoop all the marks. You don't leave any mark or any questions attended to. So that is the role of this meeting. We are going to discuss the possible revision questions. Somebody requested, and it was a good idea. But now I thought that when I go on like a video thing, it's be like a video thing, and then I read the questions and tell how to answer them. I think I will not have direct contact with the students so much. So I thought I would upload it here, and then I'll be able to explain each and every question, how we need to answer. But it's still okay. Nothing is lost. Yes, I can see still BMLS, BMS, those are medicine students, still loading. Please, let's load. We are at 100. Only a minute to 9:20, then I will start. Thank you so much. Thank you so much for the support. Thank you so much for the subscription. Thank you so much for following. We will go far. It will help a lot. If it saves a soul who has been feeling that biochem is difficult, then that is the role of this channel and the role of these meetings we are holding live. We need to prepare ourselves since next week will be a week of exams. So we need to prepare ourselves on how to answer. If any of the questions come in exam or in the CAT for my BMS students, medicine students who are having their CAT on Tuesday and Wednesday. And all the best with the preparation for exams and for the CATs as well. So I'm almost starting. We are almost starting. We are at 111. Very good number. Thank you so much. Yes, BMS, we are still there. I can see Jenny representing the BMS. Stella, I'm not seeing you. Did you already log in? Karen, are you there? You can just send a message. The class reps, Kevin, Victor, who did I leave behind? Asfa will always put me on toes. So I always appreciate. I didn't even know that I had to send a link. I thought immediately I'm live, everybody will be able to see me, but I was wrong. But I appreciate each and every effort of everyone. I appreciate big time. So we are at 108, not bad. I'm just opening my computer so that we can start. So make sure you have a pen and a book because I'm going to explain. I read one question. We are going to start with the carbohydrates, possibly the biomolecules. So we are going to look at the structures. If a question is asked, what should we be able to think of answering? You don't take too much time answering something which is not asked. So especially for the short answered questions. So I have to make this one clear for the medicine students. Our cut, I mentioned, it will be 2 hours. And in our cut, I said the MCQs, I separate them as cut 1 and then cut 2, still on the same paper. Cut 2 are short answered. If it goes too much, it's 10 marks. It can be, but it's not beyond 10. There are 7, there are 6, there are 4. But we'll be having biomolecules on Tuesday from 9 to 11. So don't get confused thinking that we'll do cut 2 after cut 1. No, the same paper. Whether you want to start with the MCQs, it's okay. Or you want to start with short answers. As long as that one paper carries cut 1 and cut 2, 100 marks. I think for the cell biology, it doesn't reach 100. MCQs are 43. And then this other one is 45. But for the biomolecules, it gets to 100. 30 MCQs, which is cut 1, 70 short answered, cut 2. So that's how I'm going to separate the marks. So don't get confused, and we are going to have it another day. I think that is clear. So for the ones who haven't come or who are still coming, we need to start because we need not to delay the people who came a little bit early. So just as I say, we need a paper and a pen, and we need to note how we need to answer such questions because I can't upload it here. In Teams, it's easier for me, I can upload, but I still appreciate nothing is lost. We'll be able to revise. So don't get bored. Huh? Some can't join via the link sent. Oh, Salat. Sorry, I didn't see that. Sorry for that. Why can't they join? Do they have Gmail? Distribution of marks in cards? Abdi, Mohammed, are you BMLS? Shama, welcome. Abdi, Mohammed, tell me, are you BMLS? So that I can explain. Or generally, I'm going to read a question and I say 4 marks, 5 marks. Abdi, Mohammed, you are talking about distribution of marks in cards. Which one? Victoria, you are there? Abdi, yes, BMS. So which distribution do you want to know? In biomolecules, I've explained that cut 1, maximum number. Oh no, it is saying maximum number has reached. So what should I do? Can I extend it? Abdi Salat, And it says the maximum number has reached. That is why one can't join. Good. He has given me an option, but it's okay. One can search your YouTube and join that way. Thank you so much. Now we are at Mohammed Abdi Mohammed's question. Abdi Mohammed is BMS, that is medicine. You are saying distribution of marks in cuts. I've said the MCQs are going to be cut 1. And if they are going to be cut 1, the MCQs of biomolecules up there, I've given instruction, answer all questions. There is space for registration number, you write clearly. There is space for what? For name. Then I say cut 1, MCQs, cut 1, in bracket, 30 marks. Then for cut 2, those short answered questions, there is a question which at the end of the question in bracket is the number. Tab. The screen. Lian, which language is this? I don't understand. Tab, the screen. Lian is saying something. I don't understand. Tab. But let me answer Mohammed Abdi first. So, Mohammed, I'm saying in our cards for the medicine, we have cut 1. I say MCQs, cut 1. For the biomolecule is 30. For the cell biology, it is 43. So the marks which you'll get for the MCQs is for cut 1. The short answer, at the end of each question, there is marks in bracket. So you'll know the level at which you need to tackle the question. You don't go overboard. Then before you can finish, then we say time up. Then you haven't even finished. You know, the ones which have 10 marks needs a lot of explanation. And we'll see the best example match in biomolecules. So I'm going to read a question, and then in bracket I say this type of marks or how many should that one carry. Then I'm going to tell you how you need to answer it. Abdi, Mohammed, can you tell me whether I try to answer or did I go opposite? I'm trying to open my slides. For my BMLS upgrading, I appreciate your presence. So we are going to have our cut that same week when we are having the exam. So when we are having the cut for the BMS, yours is on Thursday. So as I already mentioned prior, we are going to have cut 1, 20 marks, cut 2, 25. The same paper, 1 hour. For my nursing students, regular, because we are all represented here, I need to make things clear. So for my nursing students, regular, I said that the first cut which already did was 70 marks. So the cut 2 will only be 10 marks and it will be for 20 minutes. So that will be on Tuesday. That is how we agreed. So I've made it clear for everyone. So it means the cuts for the nursing students, you need not to think a lot. It's still biomolecules. What I stress much on biomolecules, do you know the structures? So the structures of the monosaccharides. Can you draw the linear form? Can you draw the cyclic form? If somebody talks about an alpha D, which form should you draw? If somebody talks about only L galactose, D galactose, can you draw it? For my nursing and my BMS students, I tell you to derive the Henderson-Hasselbalch equation or the equation used in preparation of buffers in the lab. Can you do that? Can you not confuse the KEQ and KA? So just a few points, you put them in order. Do you know how to draw a peptide, be it tripeptide, be it tetrapeptide? How many water molecules are released? Those are what we are trying to look at. Nothing much. So don't overthink. Don't overthink. What I teach is what I set. So Mohammed Abdi, can you tell me if I answered what you wanted or you had a different question? I'm opening my questions tab. on my computer, so I'll be able to read the questions. Please don't get tired. Don't get bored that Madam is now not projecting. It is going to help a mile. Even this one, if you come to understand the recordings, even while revising tomorrow, it will be of help because you'll try to recollect what Madam said concerning how we can answer. So let me open my questions and we can start with the carbohydrates, the possible questions. But still, if the site can allow me to upload them as a Word document, that will also be okay because it doesn't accept the Word document. So uploading them becomes difficult. What I'll do, I'll also try to explain how those ones can be answered, like a video. Then you'll also be able to pick. And together with whatever we are discussing now, then it will be of help. So we can start with the carbohydrates because buffers and water, not much, even nucleic acids, not much. So if you start with the carbohydrates, we are going to start with the short answered in the carbohydrate section, short answered in the amino acid section or the protein section, short answered in the lipid section, and then we shall revisit the questions. If anybody has a question, you can do the chat. I don't know whether somebody can speak. Are you allowed to speak from your end so that I can hear if somebody has a question, or you'll just chat and then I'll be able to answer either way. Then we look at the essay questions. So how you need to answer when you see such a question so that you don't answer your own things. For example, if somebody talks about naming of monosaccharides, discuss based on the functional group. And then somebody didn't understand it well, try to define much of the functional isomers and stereoisomers. No, that is not the thing. So that is the essence of this meeting. We need to understand how and where we need to put our points right when answering such questions. So I hope it will be of help to all the students who have logged in and even the ones who are still on their way to log in or the ones who have already logged in the channel. So it is still okay. So let me open and we start with the carbohydrates. Mohammed, I'm still waiting. Did I answer you? BMS, Mohammed Abdi? Did I answer you well? You talked about distribution of marks. Leon, tab the screen. Tab the screen means what? If I tab, do people join or the ones who are stuck can come? Leon hasn't answered, and I'm waiting also for Mohammed to say something. But then if they don't have anything to say, even to respond, it's okay. We shall proceed. It's just opening at 100%. I have to wait for it to open so that we can move together. But then I still thank you so much for dedicating your time to log in so that we can learn together. It is a step for all of us. It's a journey which involves all of us. And I'll be happy at the end of the semester when all my nursing students have passed, when all my BMLS upgrading have passed, when all my MBCHB in July next year have passed. Because if they do well in the CATs, then it determines that the end exam will not be difficult. So it needs a support, everyone's effort. Did I open? I think it's opening. Yeah, I think the number is fine. It's going at 137, 132. I think the ones who can't join live, as Salat suggested, they can go to the channel and still can join. So we are at the carbohydrates. the short answered questions. So question number one says, if you can hear me, if you can't hear me, work. Did you come back? What is he called? Work check. Did you come back, please? If you can't hear me, just log out and log in again. So we are at carbohydrates. We are going to discuss the short answered questions, how we need to answer them in any case you see those questions in exam or in CAT. How do we need to answer them? So we are at carbohydrates. And point number one says, draw the structure of lactose. Draw the structure of lactose and state the monosaccharide components and the type of glycosidic bond formed. Draw lactose. State the monosaccharide component and the glycosidic bond which is forming there. In bracket, 4 marks. Go straight to the point. Draw the structure of lactose. Lactose has a component of beta-D-galactose as the first monosaccharide component. Beta-D-galactose. The second monosaccharide component, beta-D-glucose. And you know very well, galactose at carbon atom number 4, it is an epimer of glucose at that carbon atom number 4. So it means we have beta-D-galactose. That already tells you it has to be cyclic. So the first monosaccharide component is beta-D-galactose. And we said for beta, at carbon atom number 1, the OH is above. And then, since it is galactose, at carbon atom number 3 and number 4, the OH will be in the same position. So it means the OH is inside at carbon atom number 3. Then it means at carbon atom number 4, it is above, meaning it is to the left. So the first component of lactose has to be beta-D-galactose. And that is how I see. If I see it is not galactose, then the whole structure is wrong. Here, the structure of lactose will get us the 2 marks. 2 crude marks. Then the beta-D-glucose, the second component. But since it is above at carbon atom number 1 of beta-D-galactose, it will extend. The glycosidic bond will extend upwards because the OH is above. It is beta-D-galactose. So it will attach itself at the first carbon atom of beta-D-galactose and the fourth carbon atom of beta-D-glucose. So the type of glycosidic bond forming there is beta, 1,4 glycosidic bond. That is the third mark. The fourth mark will be, you have to specify. Don't just say galactose and glucose. I'll not give you a mark, even a single one. State beta-D-galactose, beta-D-glucose. 4 marks. That easy. Don't explain that it is a lactose, it is a milk sugar. No, that one you are not asked. That one come and explain in disaccharide, in details. But in this case, you are asked about draw. It is short answer. It tells you straight, draw the structure of lactose. Draw the structure. Mention the type of glycosidic bond. 1 mark, beta-1,4. Go straight, mention beta-D-galactose, beta-D-glucose, full stop. Don't go to stories. Go to the next question. Don't write a lot of stories here. In essays is where you are told discuss. Discuss in length disaccharides. You will discuss all the stories, milk, sugar, lactose intolerance, yes. But here we need the structure. We need the monosaccharide component, which have to be specific. We need what? We need the type of glycosidic bond. Question number 2. Define epimers. Define epimers. And give structures of 2 monosaccharide components to support the definition. I've said two. I did leave it open that describe epimers and give structures. I've said two. It means definition, monosaccharides. They are of the same molecular formula, same functional group, but there is a change at a specific carbon atom, which is not the carbonyl carbon. 1 mark. I've said two. The reason why I've said two, I want you to give me either glucose and galactose or glucose and mannose. Drawing the structure of D-glucose, another 1 mark, 2nd mark. Drawing the structure of D-galactose, another, the 3rd mark. Then mentioning that at carbon atom number 4 of D-glucose, it is an epimer of D-galactose is an epimer of D-glucose at carbon atom number 4, not the other way around. Don't say that glucose is the epimer of galactose. No, that was a mistake, which I saw in some students when they did the cut. So we don't make that mistake. You say D-galactose, it is the epimer, is the epimer of glucose at carbon atom number 4. That is how the 4 marks get complete. Or you can give the structure of D-glucose and D-mannose. But even if you decide to give the 3 structures, it means one of them I will not mark. You are still fine. You are, as long as you draw the correct structures. You have the aldoses, the aldehyde group there, the 6 carbons. If you are drawing the galactose, the glucose, and the mannose, even ribuloses and the xyluloses, those are also epimers. But we are not talking about those ones. The ones which you know much, which you can't forget the structures, are the ones which we've done in class, like the galactose, D-galactose. If it is D, it is D. If it's L, it is L. Don't draw for me D-glucose and L-galactose and you say they are epimers. No, that is wrong. If it is D, it is D. D-glucose, D-galactose. D-glucose, D-mannose. That is how the 4 marks gets. 1 mark definition. 2nd mark for D-glucose. 3rd mark, D-mannose or D-galactose. The last mark is to mention the carbon where they are epimers. Point number 3. This one has only 2 marks. Draw structures of a ketotriose and a ketohexose. I'm telling you, I'm always very lenient in my setting. Very lenient. There is no way you can miss and you get a 0 if you are in class. And that's why I say attendance. Attend class 100%. You won't fail and you won't miss something to write in my exam, that is. So the ketotriose, we are still back into naming of monosaccharides based on the functional group and the number of carbon atoms in the main skeleton. So if you look at the structure of ketotriose, the functional group, it is a ketose. At carbon atom number 2, we have the keto. But now triose, 3 carbon atoms. So you are drawing dihydroxyacetone. Then for the ketohexose, the one which you already know is fructose. Just draw the fructose structure. It will give us 1 mark. So the 2nd carbon is a keto. For D-fructose, that is, we said 6 carbons down the line. For D-fructose, it's an exose. The 1st carbon atom is CH2OH. The 2nd carbon atom is C double bond O, where the keto is. For D, the OH at the 3rd carbon atom, for D-fructose, the OH is to the left. Balance with the hydrogen. Characteristic of water. Ideal biological solvent. Then carbon atom number 4 and number 5, the OH is to the right, balance with the hydrogen. The last one, number 6, is CH2OH. That's how simple it is. Then you get your 2 marks. So each one of them is going to give us 1 mark, 1 mark each. So that one was reading structure of ketotriose and ketohexose in brackets, 2 marks. Question number 4. We are still on carbohydrates. Short answered. Any can come. So question number 4 says, show structural similarity and difference. Show structural similarity and difference between alpha L glucose and beta L galactose. Structural similarity, no stories. I told you my exams in biomolecules is always knowing the structures of the monomers. Very important. So here, if you draw the structure of alpha L glucose, the examiner is saying draw your structure in Howard projection. If you see alpha and beta, Howard or cyclic form, draw it in cyclic form. If you don't see alpha or beta, draw it in linear form. Simple. So the similarity in structure here is both galactose and glucose are aldoses. So they are going to form a pyranose, the six-membered ring. Draw it correctly, because if they are aldoses, automatically, when the OH at carbon atom number 5 forms the hemiacetal with the carbon atom number 1 functional group, it is going to form a six-membered ring. Go straight, draw the six-membered ring for both the glucose and the galactose. The difference here is, as much as you will see the L-galactose now, the OH at carbon atom number 3 will come outside the ring. At the same position, at carbon atom number 4, it will be below the ring here. It will be the same position. But another difference which we'll note is the beta has the OH above, the alpha will have the OH below at carbon atom number 1. So just go straight and draw your structures. No stories. Don't explain. I told you when you learn the structures, you'll know the secret of passing my exams. They're very easy. If you know how to balance the bonds around those atoms, be it carbon, be it nitrogen, very simple. So in this case, just draw the structure. Don't even go to a column and explain to me the difference and similarity. I'll see it from the structure. So do you know how an alpha should look like? Do you know how a beta should look like? Do you know how to draw the L? Do you know how to draw the L-glucose and the L-galactose? That is how simple it is. Question number 5. Define pyranosis and furanosis and one structural example to support your definition. Define pyranosis and furanosis. Simple. Pyranoses are the six-membered ring. Simple. Furanose, five-membered ring. Simple. You can draw the pyran and the furan without even indicating that it is glucose or fructose you are drawing. Just get your mark. So draw the five-membered ring, draw the six-membered ring for pyranoses, and then the five-membered ring for the furanoses. That is how simple it is. Go straight to the point. The way it is asked, just go straight. Draw the structures. Don't explain anything. Where I need explanation, I'll let you know. Like discuss cellulose or discuss starch. Those one need stories. So in this case, just by drawing the structure, you'll be able to differentiate the two. It doesn't mean that it has to be the exact thing, but somebody can even ask for fructose, beta L fructose, alpha D mannose. So just by looking at the example, you shall be able to know this is how I need to answer this question. Number 6, draw two structures in each case to define and differentiate between epimers. So we are defining epimers, the same question as the first one. The same question as the first one, which we answered in number 2. So you are defining epimers. After defining epimers, you can also give the structure of D-glucose and D-galactose, or D-glucose and D-mannose, bearing in mind that the last mark, the fourth mark, will be when you specify at what carbon are they epimers. So it is similar. The way you answer question number 6 is similar as question number 2. So question number 7 still says, use a structural example to differentiate between alpha L mannose and L galactose. It's saying, structural example to differentiate between alpha L mannose and L galactose. What I'm testing there, can you draw the linear form? Can you draw the Howard projection, the Fisher's projection? So I've said if they talk about alpha, it has to be in cyclic form. And the alphanicity and the betanicity of those sugars when drawing them is determined by carbon atom number 1. So at carbon atom number 1, the OH is below the ring for the L mannose. But remember now, it is L mannose. So at carbon atom number 2, instead of the OH being inside the ring, it will be outside the ring, together with carbon atom number 3, because it is L mannose. But it is alpha. The OH at carbon atom number 1 will be below the ring. For the L-galactose, you are drawing a linear form. Without the beta and alpha, draw the linear form, the Fisher's projection. So for the L-galactose, you will draw 6 carbons down the line. It is an exhaust. At carbon atom number 1, you will have double bond O and a hydrogen, an aldehyde group. At carbon atom number 6, it will be CH2OH. But at carbon atom number 3 and number 4, since you are drawing L-galactose, the OH at carbon atom number 3 and number 4 will be to the right, balanced with the hydrogen. It means that carbon atom number 2 and number 5 bring the OH to the left, and that is how L-galactose will look like. Question number 8. Define functional isomers and give structures of 2 monosaccharides to support your definition. So if you talk about functional isomers, if you talk about functional isomers, it means they are of the same molecular formula, but the difference is based much on the different functional groups. So if they are exoses, they are C6H12O6. But we have one which is an aldose, that is glucose, D-glucose for that matter. At carbon atom number 1, you have the aldehyde group. Then you compare it with fructose, which is a ketohexose. At carbon atom number 2, you have a keto group. So those are functional isomers. Another example could be ketotriose and aldotriose. We are talking about glyceraldehydes and dihydroxyacetone. So one is a ketose, the other one is an aldose. They are functional isomers. We said stereoisomers, as much as they are of the same molecular formula, but they are mirror images of one another. Example of stereoisomers are like D-glucose and L-glucose. So we need to make these things clear. Whenever you are told to draw structures, don't write stories. Just go straight there, that is 4 marks. Draw the structure of glucose, draw the structure of fructose, define functional isomers, those are 3 marks, and then mention that fructose is a keto, glucose is an aldose, or it has an aldehyde group. 4 marks. Just go straight, be brief, and then you can move to the next question. Question number 9 says, it still talks about lactose and state the monosaccharide component. I think it has just been repeated. But we can also discuss maltose and sucrose. So sucrose, if you're told to draw the structure of sucrose, most students will always confuse the second carbon, putting an O oxygen there. It should not be OH for sucrose. Remember, it is a beta D fructose. The absence of oxygen, it means all the monosaccharide components are involved in bonding, and that is why sucrose is non-reducing sugar. So when you are drawing the structure of sucrose, the second monosaccharide component, that is beta D fructose, should not have the oxygen. It should only be hydrogen. And some students even shift the CH2OH at carbon atom number 6 by bringing it inside. No, it doesn't affect the sixth carbon. It remains CH2OH. Only at carbon atom, which is here, where the OH is above, there is absence of oxygen on fructose. But the monosaccharide components are alpha D glucose and beta D fructose. The type of one, then you draw the structures of the monosaccharide component. For maltose is the simplest. It has alpha 1,4. The first glucose component is an alpha D glucose. The second one is a beta D glucose. At carbon atom number 1 of the alpha D glucose, it attaches itself with the fourth carbon atom of beta D glucose, and it forms an alpha 1,4 glycosidic bond. So because the question was reading lactose, it already found when we discussed the lactose, like the first question on the carbohydrates, and that is why I changed now to discuss sucrose and lactose. We are on lipids. Somebody with a question on the carbohydrate, or we can finish fast. The lipids, that is the fatty acids, the buffers and water, nucleic acids, or I can finish fast and then somebody can ask question if there is. So we are on lipids as biomolecules. And still, I repeat by saying that when you work on the biomolecule, as fast. So that's why we've been discussing much of the monosaccharides, the disaccharides, because they're the ones which build into the polysaccharide. It doesn't mean that I can't bring question on the homopolysaccharides. I'm just giving a clue on what need to be discussed or what you need to answer. I can say just discuss or write short notes on cellulose, still on carbohydrates. So if I say write short notes on cellulose, you can mention cellulose. The monosaccharide component there are beta D. The type of glycosidic bond which forms there is beta 1,4. And you also have to talk much about it being the structural component of most of those plants. So we talk about wood being purely cellulose or cotton. The way we mentioned, you'll be able to get your marks. I can also tell you to write short notes on amylopectin and glycogen, for example. It's very important to mention the monosaccharide components. It is a homopolysaccharide. You have to mention the monosaccharide components based on the marks, if it is 4 or 5 marks. Monosaccharide components, very important. The type of glycosidic bond found there, whether it is branched or non-branched. For the amylopectin, you know very well that it is not that branched. When you compare with glycogen, you come across maybe 20 to 30 glucose units, they will add to the marks. So based on what you've been asked, go straight to the point and discuss. Some students always confuse the cellulose as the monosaccharide component found in human beings. Some people also confuse amylopectin with glycogen. So we have to be precise when we put our points down so that we can scoop all the marks. So at first, you need to know, is this cellulose a carbohydrate? Some people will even mix a peptide bond is found there. And then you are wondering, do they know that it is a carbohydrate? So such mistakes, those small mistakes are the ones which always cost us. So it can be asked in different ways. It doesn't mean that I can't set any from the polysaccharides. Even the heteropolysaccharide, the medicine students have done a heteropolysaccharide. I can even ask about the hyaluronic acid, the medical importance of hyaluronic acid. You know very well how it contributes much to the soluble part, that fluid part. So at old age, at repairing of the tissue. So those are the information which you have to be on point. If I talk about medical and biological importance of HA, hyaluronic acid, or dermatan sulfate, just mention it. But then we don't even forget about the dextrans. Yeah. So we should always be precise with our points. You mention the monosaccharide components, the type of bond, it will always add marks. So just to show us what we need to answer in any case we come across such a question. For the lipids, among fatty acids, the first question says, define mono unsaturated fatty acids and polyunsaturated fatty acids and use structures to support your definition. 4 marks. Define monounsaturated, polyunsaturated, structures to support. 4 marks. Definition of monounsaturated, they have one double bond or at most one double bond. 1 mark. Polyunsaturated, at least 2 double bond. 2nd mark. Monounsaturated, you've been told to draw structure, not even the common name. Draw structure, define. So the monounsaturated, you can draw palmitoleic, you can draw oleic. 18:1 delta 9, 16:1 delta 9. Draw any. Polyunsaturated, we have several. 18:2 delta 9 12, 18:3 delta 9 12 15, 24 delta 5 8 11 and 14. Polyunsaturated. So we have linolenic, we have linoleic, we have arachidonic. You can draw any. Just go straight to the point. Draw the structure. Correct structure. Carboxyl terminal end to the right, methyl terminal end to the left. Remember, there are options in the channel how you can draw them using the lines, using the summary, which is in the table, using the long one, which any beginner can know. So just balance the bonds around the carbon. When there is a double bond, make sure you balance the bonds. You are good to go. Define. Oh no. Differentiate between unsaturated and saturated fatty acid. That is the second question. And give a structural difference of the same using one example each. So unsaturated, there is a double bond. They didn't say that you go further and expand mono and poly. No, that is your own. Just define unsaturated, double bond present. Saturated, no double bond. Simple, you get your 2 marks. Draw structure of any of the saturated. It can be C14:0, C12:0, C16:0, C18:0. Those are saturated. We said the colon will separate the number of carbons with the number of bonds. So there are 0 double bonds. So in that case, it reads the saturated fatty acid. Draw any of those. Even arachidic, the 20:0, it's still okay, as long as you draw a structure which is known. It has a common name. It has a structure. It has a position of double bond if there is. Then for the unsaturated, there is double bond present. Simple. Stop it at that. You can draw the structure of any monounsaturated or polyunsaturated. Go to the next question. That's how the 4 marks come about. Point number 3, question number 3. Clearly show the structural difference between palmitoleic, linoleic, oleic, and linolenic. Just structures. Palmitoleic, oleic, linoleic, and linolenic. We are talking about unsaturated fatty acids. Palmitoleic is C16:1 delta 9. Oleic, C18 colon 1 delta 9. Linoleic 18,2 delta 9 and 12. Linolenic 18,3 delta 9, 12, and 15. So you will be able to scoop all your marks. Each structure, 1 mark. I didn't say define. I said using one structural example. No, I said structural difference, sorry. Show structural difference between palmitoleic, oleic, linolenic, linoleic. Full stop. Just draw structures. Go your way. You'll get your marks. Point number 4. Using one structural example in each case, define between a monounsaturated and polyunsaturated. 4 marks. It resembles the one which we've just done in number 2. But now this one is, in fact, it is number 1. So it defines, you have to define the monounsaturated, at most 1 double bond, polyunsaturated, at least 2, then give structures. 4 marks. Question number 5. So it tells you alset. Alset, fatty acid structure. Alset, structure of those monosaccharides. Alset. Alset. So it helps you to know the setting pattern, how you need to answer. Maybe you are going a little bit further while answering, but now you can narrow it down to whatever is asked. So point number 5 says, write short notes on unsaturated fatty acids and give any 2. Write short notes. I think the write short notes might confuse students. It is good to define the unsaturated. Here I was given 1 mark for the unsaturated because it's 4 marks. Then 2 structures, already 3 marks. Then if you give me the common name or even the position of the double bond, you'll get half-half. That is how the last mark was being scooped. So it still talks about the saturated, the unsaturated. Can you define them? Do you know a structure? If you see even in the MCQs, C18 colon 9, 12, 15, can you tell the common name? Can you draw the structure if you find it is short answered? If it's an MCQs, you'll be asked whether it is any of those common names that exist there in the questions. So do you understand the structure of the fatty acids? Then you go to the short answered, you need to draw the structure. Can you tell of the carboxyl terminal end, the amino terminal, the methyl end, and also can you place the bonds correctly? Remember, I told you when you are placing the double bond, count from the carboxyl terminal end. If it is delta 9, after the 9th carbon atom, put a double bond, balance with the hydrogen. If it is delta 12, after the 12th carbon, put a double bond, balance with the hydrogen. That's how simple it is. Count from the carboxyl terminal end. The omega-3, omega-6, and omega-9s come about after you place the double bond. So from the methyl end, the first carbon where you meet the first double bond. And that is why the linolenic will be omega-3, the linoleic will be omega-6, and the oleic will be omega-9. After placing the double bond, from the methyl end, what carbon or which carbon number do you come across the first double bond? That's how simple it is. But it's very important to know the common names. The linoleic, the laurate, the myristate, the palmitate, palmitoleic, very important. And it will be able to be very easy to answer. The last question under lipid says, use structures to differentiate between monounsaturated and polyunsaturated and state the common names of the drawn structures. So here, you are not defining anything. I'm not giving mark for definition. It's still 4 marks. Just draw structure of any one monounsaturated. Draw structure of any one polyunsaturated. Give me the common names. you'll get your 4 marks. Those are the possible questions which come in lipids, the fatty acids. Knowing the structure, the common names, how you can draw those double bonds. Very helpful. I didn't introduce the lipids, the phospholipids, the glycolipids. No, those are things we can read and understand. The bit which is difficult to understand is what we are trying to capture. Can we know how to draw structures such that when it comes to metabolism for the medicine students in semester 3, for the nursing students in semester 2, for the BMLS, they're still not doing metabolism, you'll be able to understand how the metabolic pathways, degradation of those simple monomers, which we learned in biochemistry 1. What is the importance of that metabolic pathway which they are involved in? How important the metabolic pathway is? Because we are going to be told, describe a pathway using structures and the correct enzymes. So we have to learn the structures here in biomolecules. When we reach glycolytic pathway, you know how a D-glucose looks like and how we tackle the specificity of enzymes for the BMLS and the nursing, the substrate specificity, optical specificity, you'll know in glycolytic pathway, we are only dealing with D-glucose. In degradation of amino acid, we are only dealing with the L's. Then BCHB will be able to learn it next semester in enzymes. So that's how important it is to start with the biomolecules. We have to dwell much on structure, my friend. My questions, almost 90% is structures. We have to know the structures, very important. It helps us to know much the reactions which are going to take place during the metabolic pathways. You'll see how important those structures are when degradation takes place, the final product which is generated from those pathways, how they help for the other pathways. So in bioinformatics, we talk about interatomics, the interaction between the omics. We have the genomics, we have the proteomics, we have the metabolomics. So the metabolic pathways connecting to the proteins, connect to the genes. Because at the point you're talking about a gene product, you're talking about the protein itself, which has the amino acid in it. How they are connected. Still, the carbohydrate metabolism is connected to lipid metabolism, connected to protein metabolism, but it starts from the biomolecules. So for water and buffers, I don't have much questions here, but a bit of, in the short answered questions, of course, we can touch on, write short notes, any of the characteristics of water. It could be ideal solvent, or it could also be water as a buffer, and maybe write an equation where water is involved as a buffer, where we have the two molecules of water giving us the hydronium ion and the hydroxyl ion. So any of those characteristics of water, for example, here I talked about write short notes on crystal structure of water, 4 marks. Write short notes on cohesion and adhesion forces. Remember how well they also help in plants for water to flow from the roots to the leaves. So we talk about the capillary, So just a bit of the characteristics of water. For deriving the pH of water 7, it has to come in the essays because it's about 10 marks. It can't be less than 10. So it has to come in essay. It can come in essay, or for my medicine students, I can set it in a short answer. I still mention it as short answer, but based on the marks, it has to be 10 marks. So I can mention any of the characteristics of water in the short answer, maybe it's 4 marks or 3 marks. And then there is this long question which you need to take your time because you need to derive the pH of water 7. And you don't need a calculator anyways, because you already know it after calculating. It's the same value which we are going to get. So you don't need of having the calculator, even on that Tuesday. Then you'll be telling me, Madam, I'm waiting to calculate. No, it is not general chemistry where you are going to calculate the concentration. This one is something which you already know. So that day, if you have a calculator, you are going to put it inside your bag because I'm not going to need it. I don't know whether you have a Mwakenya there in the calculator, because nowadays the students are very cunning. They can draw it using a pen which doesn't write. And then they're busy telling you, I'm going to calculate. Nothing is going to be calculated there. Because deriving the Henderson-Hasselbalch equation also do not need a calculator. So those are the equations which can come. For the Henderson-Hasselbalch equation, it can come in the short answer because it has to be 10. I can't put it less than 10. Maybe if you have to define buffer capacity, buffer solution, buffer range, that one can go into short answered questions. And then maybe still factors to consider when selecting buffers can also come in the 10 marks. But there isn't a lot of questions for water and buffers. Mostly I don't set on the acids and bases because I think most of it you do in general chemistry, I think. So I don't always like setting acids and bases, though it helps us to understand much on how important the buffers are, but I will not stress much on the acids and bases. So just by looking at the nucleic acids before we can finish on the proteins, that is amino acid, then I can look at the ESA equations so that I don't let you get bored so much. I thought I'll run it for 1 hour, which I'm not going bad, but then the nucleic acids, what I can ask possibly is draw the structure of the RNA and the DNA. The MBCHB students, that one you had to go and print. And then when we were discussing in class, you need to have the structure there. So you can draw the structure of RNA and DNA. Remember at carbon atom number 2, for the RNA, the OH is present. For the DNA, the oxygen is not there. But what you have to note here is they are forming a furanose ring together with the fructose. So the RNA and the DNA, those are the nucleic acids. You can draw those structures. You can also be told to draw the structure of the purines, the adenines and the guanine, or pyrimidines, the cytosine, thiamine, and uracil. That is fine. And then maybe you can write any 5 functions of a nucleotide, and you can differentiate. Can you be able to differentiate between a nucleic acid, a nucleoside, and a nucleotide? You know, the nucleic acids, the nucleotides. You know, the nucleotides are the building blocks of the nucleic acids. But you have to mention a nucleotide should have a base, a phosphate, and a pentose sugar. But if it is minus the phosphate, it is called a nucleoside. And then the nucleic acids, the DNA and the RNAs, they are the polymers of the nucleotide. That's how simple it is. So, not much questions on nucleic acids and water, but let's finish by looking at proteins, the amino acids. That is where I have a problem because I'm going to set. You can draw any of the peptides, but you can start using your name. If the 1-letter codes exist in the first name of yours, maybe we have Alice. Yes. For men, let me be fair. For men, we can have who? We can have who? Dan, for example. So you can practice using your names, and then you can know, you can tell whether it is a tetrapeptide, a dipeptide, a pentapeptide. So what do we refer to as a pentapeptide? Is it based much on the number of peptide bonds forming? Is it based much on the number of amino acids involved in the formation of the bond? How many water molecules are released? Can you draw a peptide bond? Then basing much on the carboxyl terminal end, amino terminal end of the amino acids which are involved in formation of the bond. So that is how important the amino acids are, or the proteins. I like that part of amino acid structure. I would like to know whether you know acid falls. Can you draw an amino acid structure? Can you draw well the carboxyl terminal end, amino terminal end, the alpha carbon? There was a question somebody asked on the channel that, Madam, can we draw glycine as CH2? No, that is not true. We can clearly, I think I answered it. Maybe if they didn't see it, but it's still okay. It can help the others who didn't even see it. But we have to clearly, in glycine, we have to clearly show the R side chain, the carboxyl terminal end, the alpha carbon, the hydrogen up there, and the amino terminal end. So the hydrogen has to sit quietly there, not interfering, no CH2s, no H2. I think he was talking about the hydrogen up there. We combine it with the alpha carbon and the hydrogen down here. Separate it clearly. If it is not for the branch chain amino acids, there is a point, or there are some books which will always write CH, and then they write two of the CH3s, which is understandable because it's at the R side chain. Me, as long as you balance those bonds around there, because even you can see for the asparagine, but you make sure the asparagine, there is that CH up there, because for the aspartate, there is one CH2. Then the amine and the carboxyl group. So the one in the channel is just the same as the one in the notes. What I'm looking at is, can we balance the bonds around the carbon? And in asparagine, do we have both the carboxyl together with the amine group, but then there is a CH up there attaching them? So you can see there is a CH and then there is C double bond O and NH2. The bonds are balanced around the C double bond O, that is the carboxyl, and then the amine, but there is still a CH up there for asparagine. So as long as we balance the bonds. So a question can be brought in the short answered. The dicarboxylic, diamino, you have to know the number of amino group there, the number of carboxyl group there, including the R side chain. Monoamino, dicarboxylic. So we just have to know monoamino, monocarboxylic. I'm trying to see that nobody, you can't just miss to know how to draw structures, even for simplest glycine and alanine. Then I'll be fair and I say monoamino, monocarboxylic. So based on the nutritional classification, based on the classification on R side chain, based on the number of carboxyl and amino group, based on the charge, the neutral ones, the acidic, the basic, it can be set either way, be it in the essays, the one which have longer answers, it is still okay, as long as you be precise and understand the question well. So the first question says, draw a tripeptide of LNW. I said, you have to go and practice with your name. As long as one of the letter codes of the 20 amino acids appear in your first name, just put it together and see if you can draw a structure of a peptide, be it a di, tri, tetra, the same. So LNW is a tripeptide. The 3 amino acids involved. It is a tripeptide. 2 water molecules is going to get released. So we have leucine, we have asparagine, and we have tryptophan. I said the secret. I could see some students also making a mistake when I was marking the cut. I said, draw these amino acids separately. They are going to earn you 1 mark each, such that if you don't get one of the amino acid structure right, you'll be able to scoop the 2 marks. The other mark you will not get because you have the correct peptide bond forming, but you'll get the correct marks for those amino acids which will draw individually correctly. So draw them correctly, individual amino acids, then the final mark here. So the first 3 marks go for each of those amino acids. Do you know the one? Do you know the R side chain of that amino acid? Can you clearly draw a peptide bond? That is all I'm testing when I have to tell you to draw for me. a tripeptide or a peptide bond. So in this case, the final mark will go for the peptide bond plus 2 molecules of water. So the peptide bond will get half, 2 molecules of water, the other half, you'll have 4 marks. This one is not done by MBCHB, but it is done by my BMLS upgrading and nursing students regular. Write short notes on absolute group specificity. This is where the enzymes have high order of specificity. Remember, we are talking about the group specificity where the enzymes are on specific groups. Groups, what in short or in summary is by looking at peptide bond is found in proteins, glycosidic bond in carbohydrates, and the ester bond found in lipids. So if you talk about absolute group specificity, it means there are still proteases. They work on peptide bond, but it's of high order because in absolute group specificity, chymotrypsin, one of the protein splitting enzyme, will cut the peptide bond when at the carboxyl end it sees or it identifies any of the aromatic amino acids, tyrosine, phenylalanine, and tryptophan. For trypsin, also one of the protein splitting enzyme, will have high order of specificity. It will cut carboxyl end when it sees the basic amino acids like histidine, arginine, and lysine. So we are talking about specificity of enzymes, substrate specificity, optical specificity, reaction specificity, group specificity. We should always know all those at our fingertips as we answer the essay part in the main exam for the BMLS and the nursing students, as well as at the point where you answer them in the short answered. So you'll be able to answer any. If it comes to short answered, just be precise. Some students were confusing reaction specificity and group specificity, which is not true. So for the group specificity, you go for a peptide bond if it's a protein, glycosidic bond if it is for carbohydrates, and an esterase or an ester bond for the lipids. Another question says, draw structure of cysteine and serine. I think this one I can't set for my regular students and the MBCHB. I always make it simpler for my online students because I don't meet them face to face. We meet online until we do the cut. So sometimes you try to be fair. Something which they can understand even as they read, even if you don't draw it. So for the cysteine, it was the sulfur containing, very simple one. The R side chain has CH2SH, is the one which is used in formation of the sulfur bridges. Serine was the alcohol containing, it is CH2OH. So those are the 2 amino acids. It is 2 marks, each one of them getting 1 mark. Question number 4 says, aspartate and lysine are 2 different amino acids classified in 2 different classes based on the R group or side chain. Mention the group and draw the structures. So the groups, aspartate is acidic, lysine is basic. You draw the structure of aspartate. Lysine is the one which have 4 of the CH2s and an NH2. The NH2 tells much about the basic behavior of that amino acid. Then point number 5, write short notes on any 4 classes of enzymes. This is also not covered by MBCHB, but I thought I should revise it. So if you discuss, write short notes on any 4 classes of enzymes, it's only 4 marks. So just by mentioning the ligases, the kinases, the hydrolases, oxidoreductases, and defining the reactions which they are involving will give you the full mark. So mentioning the class, half, defining another half. One of my BMLS upgrading asked me a very relevant question last 2 weeks ago. Madam, if you're asked about this, can you take 1 class and discuss in depth? No. Don't take 1 class of enzymes and discuss in depth. Write any 4. There are 6 classes according to the Biochemistry International classification uh, whatever. So in this case, you choose any four, just define why they're in that class, then leave. Don't take one. If you take one, you'll get all the marks. Just as the question says, four classes. Draw a tetrapeptide face, F-A-C-E. F, phenylalanine. A is alanine. C is cysteine. E is glutamate. Draw them separately. That is 5 marks. You'll get 4 marks for each of those amino acids because I'm trying to test. Do you know the R side chain? Can you draw their structures? Then come to the peptide bond. That is a tetrapeptide. 4 amino acids are involved. We are going to remove 3 molecules of water. Sorry. That's how simple it is. A peptide bond has to be set. A peptide bond has to be set so that we can understand all the components which are involved in amino acids and the peptides. The other one says differentiate between fibrous and globular proteins and give 2 examples. So that is classification based on the shape of proteins. This one, all the 3 groups, it's relevant. So the fibrous, just define. Globular, just define. And then give examples. You know, globular from the name, globulins are there. Just give 2. You'll get your mark. Draw structures of any 3. Monoamino, monocarboxylic. I'm trying to be considerate so that if somebody was going to get a 0, that there is nothing you can remember. At least we can remember the structure of glycine. We can remember the structure of alanine. We can remember the structure of cysteine. You can remember the structure of serine, for heaven's sake. I'm always trying to be very fair so that a student do not miss something to write. So this one is 3 marks. Monoamino, monocarboxylic. Only the one carboxyl group at the carboxyl terminal end, one amino group at the amino terminal end. There is none of the carboxyl and amino group on the R side chain. That is what they mean. So classification of amino acids based on the number of carboxyl and amino group. Draw any of those, even the branch chain amino acids, the aliphatics, the alcohol containing, the sulfur, whichever it's easier for you. It's any of those, monoamino, monocarboxylic. Differentiate between group specificity and absolute group specificity. So I said group specificity, enzymes being specific to their groups where they catalyze. It means peptidases will work on peptide bonds, glycosidases will work on glycosidic bonds, esterases will work on the ester bond. So don't confuse it with urease, which is reaction specificity. Given the lipases, reaction specificity. We are talking about the type of bond which is found on the biomolecules. Then the absolute group specificity is of a high order than the group. So just be to the point so that you can scoop all the marks. Draw tripeptide YAK, YAK, tyrosine, alanine, and lysine. Draw the amino acids individually. You get your 3 marks. The final mark is for the peptide bond and the 2 molecules of water which are released. Differentiate between uncompetitive and non-competitive inhibition types. Still, the BMLS upgrading and the nursing. We are talking about the competitive inhibitors. And I said here, by differentiating, don't write stories. Define the uncompetitive just by mentioning that the inhibitor binds only to the enzyme substrate complex. For the non-competitive, irregardless whoever binds first, no competition. But the inhibitor binds to free enzyme and the enzyme substrate, forming ESI and EI. You'll get your marks. Even if you decide to draw the reaction, it is still okay. You'll still get your mark. You can even draw that equation. You still get all your marks as long as you draw the correct thing. Write short notes on sulfonylamides or sulfonamides in treatment of bacterial infections. So generally, it touches on the chemotherapeutic use of the competitive inhibitors. For the nursing students, I gave that one as a handout. So you saw where they are involved in treatment of bacterial infection, treatment of leukemia, treatment of gout, the allopurinols, lisinoprils being treating what? Blood pressure. Speak on one. Like for the sulfonamide, I said it has sulfonylamide, which has the same structure as PABA, p-aminobenzoic acid. So they will always compete for that enzyme which is responsible for formation of folic acid. And that's how competitive inhibitors have been used much as chemotherapeutic agents. Very important. So we can pick any. The use of lisinoprils in blood pressure treatment, in blood pressure. The use of allopurinol in treatment of gout. Generally, it can come to short answer or the essay. The essay will come discuss the use of competitive inhibitors as chemotherapeutic agents. Simple. So you have to discuss all those, how they're useful in treatment of those diseases. People already going down and down, 122, but the ones who are there, we still appreciate. In the essay, I'm not going to take much time. I'm just going to mention what need to be written. So I'm starting with the carbohydrates essay because some we've covered in the short answer. I'm just going to mention to you how you need to solve those problems whenever you see them or answer those questions whenever you see them. So for the carbohydrate essay, somebody can ask a question like, using 4 structural examples in each case, discuss the 2 criteria for naming of monosaccharides. That is a simple question because I don't want somebody to get 0. In this question, I always test, can you draw the structures of the monosaccharide? Of course, the Fisher's projection, not the Howarth. I don't need it in Howarth. I need it in linear form because you'll say 1 based on the functional group. And when you mention the functional group, you have to mention to me the aldehyde group at carbon atom number 1. You get your full mark. Keto group at carbon atom number 2, another mark. Examples by names. If it is fructose, you are giving me. Here I want 4. So if you talk about functional group, you have ketose. I need 4 of the monosaccharides. I need fructose. I need ribulose. I need erythrose. I need dihydroxyacetone. You get your 4 marks. Or even if it's 1.5, you get your 6 marks. You go to aldehyde group. We are still on functional group. I need glucose. I need galactose. I need mannose. I need glyceraldehyde. Simple as that. You get your marks for the structure. Those names, when you also mention them, you get your mark. Then you go to the, based on the number of carbon atoms. Imagine, just by telling me 3 carbon strioses, you get 1 mark. 4 carbon tetroses, another mark. 5 carbon pentoses, another mark. Hexoses, 6 carbon. Then draw the structures. They'll be similar to the functional group because you'll have the 6 carbon, but then a ketose, 3 carbons, but then a ketose. You look at that. And you get all your marks. I told you there was a time I set this question and the students were writing until I could find a way of reducing the 20 so that it doesn't enter 20, even enter 19.5, but it could enter. I give all the 20 because the students have explained it well. And even the structures, you can't even reduce a half. So it shows well how we understand the question. There are questions which are like not bonga points, redeeming. not even for laser. The ones which are obvious, they will always save the situation. You look at it and you smile and you ask yourself, no, now what is Madame setting? Because they're very easy. You go straight to the point and you answer. 4 examples each. For the functional group, ketos give 4, aldose give 4. Number of carbon atoms give 4. 4 carbon, 3 carbon, 5 carbon, 6 carbon. Simple. Discuss disaccharides, including structures of the monosaccharide components and the glycosidic bond type forming. So here is where you talk much about sucrose, how it is a cane sugar. It is non-reducing because all the monosaccharide components have been involved in bonding. And then you specify the type of monosaccharide uh, components, the type of glycosidic bond, draw this, talk everything about each of the disaccharide. You get all your marks. Lactose, talk about the milk sugar, the lactose intolerance without the lactase will not be broken down. Talk about whatever is there, which we discuss in class. Maltose, the malt sugar. You see, talk about it. You'll get the 20 marks. But correctly draw those structures. You'll get full marks. But now, you know, you can't you can't set a question on carbohydrates for all the questions in section C. If it's carbohydrates, you have to put proteins, then you finish with fatty acids. But then, in each of those topics which are covered, let us try to scoop even 10 out of 20 if it's too hard. But I know if you put an effort, it shall pay back. Point number 3 says, I think it's still the same one, using 3 structural examples in each case, discuss in detail the 2 criteria in naming of monosaccharides. So that is the same as the first one. So the mistake which you don't make here is, if I talk about naming of monosaccharide criteria, don't put functional isomers and epimers anywhere. No. Oh, somebody want to join. I'm just seeing, sorry, for the ones who have been trying to join, I just see my screen showing a light, so I don't know that they were joining because I'm busy discussing. But if I see, I'll accept, please. I'll accept. Just try to apply again. I'll accept. So don't confuse by writing about the functional isomers here. We only need the number of carbon atoms in the main skeleton and the functional group. That's how simple it is. Just give the structures. Please, I always feel sad when the student had an idea, but now they come in with epimers and the functional isomers and the Haworth projection and the Fisher's projection. No, that is not what I need. Naming of monosaccharide is based much on the functional group and the number of carbon atoms. Number 4 still explains much. With reference to relevant structures, discuss disaccharides in details. So here is where we also have the last question saying that discuss the homo polysaccharides. But I think that is too wide. I can narrow it down and say discuss starch and cellulose, for example. Because starch takes a lot. Just by mentioning the component of carbohydrate found in plants, amylose and amylopectin, and diving into each of the amylose and amylopectin already costs too much. Then you go to cellulose. So it is good if somebody sets and says, discuss cellulose and starch, for example. Then you can also discuss heteropolysaccharide is what I can say there. But even heteropolysaccharide, the HA, hyaluronic, has a lot of points. So maybe I can mention hyaluronic and any of the heteropolysaccharide. So those always come in essay. You talk in length, in detail. You discuss about a certain component which have been asked in the questions. Proteins, it says, mostly in proteins, I have the enzyme, factors affecting enzyme activity. So in this case, I've said discuss the effect of the following on enzyme activity. Temperature, I put 10 marks. pH, I put 5 marks. Substrate concentration, I put 5 marks. It means in temperature, I mentioned, just as I mentioned it in class or in the handout, specify at 0 degrees, whatever happens, the enzyme is inactive. No enzyme substrate complex formed. At 20 degrees, we are increasing the temperature. The kinetic energy is increasing. There is collision of molecules. At 37 degrees, optimum temperature at which the enzyme works best, the formation of products to the highest. Beyond 60 degrees, denaturation occurs. So enzymes are protein in nature. When they get denatured, the biological activity is lost. And I said, and I still repeat, when it is denatured, a protein is denatured, which only affects the secondary, the tertiary, and the quaternary structure. The primary structure is linear, so it is not affected. Then you can draw the graph. Up there, put a title, Effect of Temperature on Enzyme Activity. A graph cannot be there without a title. To the horizontal side, horizontal, vertical. Where is the horizontal, the vertical? To the vertical side is the enzyme activity. Horizontal is that factor which you are looking at, if it is temperature. For temperature, it will have a curve which looks more like bell-shaped. Then you go to pH. And pH, I said, each of those enzymes has an optimum pH. The enzyme which digests proteins work well in acidic pH. If you expose them to a neutral pH where the amylase works, to a basic pH where the lipase work, they get denatured. Just the same way as the lipase, which digests lipids, you can't expose it to acidic pH, neither can you expose it to a neutral pH. It will denature. So for pH, it differs per enzyme. You know the graph which I showed for those. Draw the graph. All the graphs should have a title. Substrate concentration. As we increase the substrate, the enzymes, sites on the enzyme for the substrate to bind are still available. But it reaches saturation point where now the values are flat. There is no more sites on the enzymes for the substrate to bind. That's how simple it is. Give the graph, you'll get your marks. Don't write a story, a very paragraph, which makes me to look for the marks. No, life something which is brief in point form. It makes life easier for you, even for the examiner as well. And let us have a good handwriting. You know, some students have points, but the way they write, you can lose your marks. Not every lecturer will be able to read whatever you've written. Write a good handwriting which can be seen and good, but something which can be read. Because you can have points, but now you are losing it. Some people will write a 0 for you, yet you had points there because you didn't write clearly. And we're just human beings. Maybe the brain is now exhausted in marking. So maybe they can see you didn't write anything, yet you wrote something. So let us write well. Enzyme inhibition types. Those are the irreversible and the reversible, still on my BMLS, and the regular nursing. So you'll be able to discuss the reversible inhibition types and the irreversible and give the equation. I told you about the equation, the binding of the substrate, the binding of inhibitor. That's the only thing you need, and you'll get your marks. Then there is this question which talks about competitive inhibitors are used as chemotherapeutic agents. Discuss. That is different. Don't start by defining the competitive inhibitors. Just go straight and use the examples of those drugs which are used in treatment of such. Then there is a question for all the three groups. Draw structures of all aliphatic. All aliphatic. That is the simplest question. You know, in essay, for my amino acids, I always have a challenge because I just have to set it in an easy way. I will even tell you maybe based on the nutritional classification, discuss by drawing structures any of 6 non-essential, 6 essential amino acids, including their 1 and 3 letter codes. So I just have to set a question which will touch on drawing of the amino acids. So here I'm saying draw all the aliphatic, because I know you can't miss to draw alanine. You can't miss to draw glycine. You can't miss to draw valine. You can't miss to draw leucine. Maybe isoleucine, but it's also not difficult. Proline, the two versions of proline are still okay. The one in class looks a bit simple because of the CH2s, three of them, which we haven't included. But in the channel, it is the same structure, but now the CH3s, the CH2s, sorry, are connected, the three of them. So those are the aliphatics, the proline, the branch chain, the alanine and valine. And then the sulfur containing are methionine and cysteine. Include 3 and 1 letter code. Imagine, you can get a 10. 1 letter code of the aliphatics, they are just from the 1 letter code. Then the 3 letter codes, they are obvious. So I'm trying to be lenient so that the student won't miss something. Discuss protein classification based on solubility and shape. So that is also what you need to write notes on. It doesn't need structures. So you'll talk about the solubility, the ones which are soluble in alkali, the ones which are soluble in water, the ones which are insoluble, and then you'll come to the shape, the fibrous and the globular proteins. I know people are tired. We are just human beings. Some people have already gone to sleep. Tired. Madam is talking too much. I'm talking about another question. Any 5 structural examples in each case? Discuss nutritional classification of amino acids. So you have to define the essential and the non-essential. Then another question talks about specificity of enzymes. That one we already done, the substrate specificity, absolute specificity, the reaction specificity, as well as the optical specificity. In lipids, just one question to catch up on the same. You'll just be told, discuss by defining the structures of 2 saturated, mono unsaturated, and polyunsaturated, including their common names and position. By position of the double bond is, you have to tell me clearly C18 colon 3 delta 9, 12, and 15. Imagine, and you get your mark. Common name, tell me linolenic. Imagine, you get your mark. Drawing the structure, that is what I can test in the essay. I'll have to accommodate the saturated. Do you understand the saturated? Can you define it? Can you draw structure? Monounsaturated, polyunsaturated. That's how simple it is. Don't stress yourself. Water and buffers, I already mentioned this, by deriving the neutral pH of water 7, given a KEQ value of 1.8 times 10 to minus 16. I already did it on the channel. So for the regular group and the MBCHB, kindly you can revisit the channel for the MBCHB before our cut on Tuesday. If you see that question, then you should be able to answer it, because I had to write it on a paper and do a picture, then upload. That is the bit I could do. So that I thought possibly some students didn't get it right, because by then some students were still registering, some were still not there on time. So now it's on the channel. So you have to derive the pH of water as 7. Remember the rules. Hydrogen ion, the subject, introducing logs, introducing negatives, define very well. pH negative log of hydrogen ion concentration substitute. The ratio, 7:7. Remember, it is KEQ for dissociation, constant of water. Then you come to Henderson-Hasselbalch equation. It has to be Ka. You are dissociating Ha to hydrogen ions and A minus. A minus is the conjugate base. Ha is the weak acid. Those are the components of buffer. But remember, pKa, negative log Ka. pH, negative log hydrogen ion concentration. Don't confuse KEQ and Ka, because when you are deriving KEQ and you put Ka, you will be stuck somewhere where now you see things are not entering. Ka is for the weak acid, for the Henderson-Hasselbalch. KEQ is for water. I think we are done. Nucleic acid still, I can set, give the difference between RNA and DNA. You remember the columns for the MBCHB students, I told you to. Did I tell you or we did it in class? But then the question which will follow that, for it to add to 20, there has to be structures of the DNA and the RNA. Thank you for your time. I hope those one will be helpful to the students. It's already 11 sharp. Almost 2 hours. I never stay that long. Some story for staying long. But I thank you for the ones who have been with us until now. For the ones who have left, I can understand. Maybe the bundle, maybe people are tired. We are just human beings. I can't sacrifice you. But then at the point of you've already logged in, I appreciate. So is somebody asking a question? It is saying maximum number has reached. Oh, these were the last time. Do somebody have a question? If no question, then we will leave. Do we have questions? How can I see the questions? Or people are tired? I'm talking to myself. Are you there? So can somebody say yes? Or the screen of chatting is full until people can't write? Is the chat room full? Okay. So let me conclude or put a closing what? Closing remark. Thank you so much. And I wish you well as you prepare for the CAT for my MBCHB. You know, your CAT looks more like an exam on Tuesday and Wednesday. For my regular and BMLS upgrading, I also wish you well as you prepare for the CAT 2 for the regular nursing, as we prepare for CAT 1 and 2 for my BMLS upgrading, and as we prepare well for the main exam, which starts on 9th. So may God bless you all, and we have a good night. And